Hello guys, so today we are going to look how to study different type of staining technique in a very interesting way. So today we will be focusing mainly on the gram staining. Before we move on to gram staining, I would like to add an introduction to these staining techniques. So, why are we doing staining techniques? This is because a normal a bacteria cannot be easily seen under the light microscope without a stain. So, we are staining this bacteria and then we are observing this in the microscope. So before staining, before doing anything, imagine you are going to write something in a paper. Okay, imagine you are going for your exam, you bring your exam board or in notice board you put a paper and then you put a paper pin on that, right? So this is why it has to stay in that place, right? So before we are staining, the smear are fixed in the slides. So fixation will also protect the internal structures of the cell in a given position. So there are two ways by which you can do this fixing technique. First one is heat fixation and second one is methanol fixation. So heat fixation is done by using heat, and this is normally done for bacterial smears, whereas in case of methanol fixation it is used for blood smear. Now there are different type of staining techniques that we use. So one is the symbol stain, where you use basic dyes like methylene blue or basic fusion. But the problem with this that every bacteria, all of them are having same color. See, all of them produced the same color, so we cannot easily differentiate which bacteria is which. Okay, so all of them are having same color. But in case that's a kind of a negative point to this symbol staining. Now the second method we can use is a negative staining. So how will you do a negative staining? You are taking a drop of the bacterial suspension and then you are using some stains like India ink or necrosin. India ink or necrosin. So what will happen? See, the background, it got colored with black in color, but the bacterial capsule refused to take up this India ink or necrosin. So it will get unstained, and that will stand out. So when you use negative staining, you are using India ink or you're using necrosin. What will you get? You get a black background with capsule. This is the black background and this capsule is unstained. This portion is the capsule and that portion is unstained. Capsule gets unstained. Next is impregnation method. So what is impregnation method? Normally the bacterial cell or its structures are very thin. You cannot normally see it under the light microscope. To make the structures a little bit more thicker, we are impregnating silver salts on their surface. So this bacteria, very thin bacteria, it is taking up the silver stain, and then silver salt, it took up the silver salt and it become big fat lady. Okay, that is impregnation method. Now the differential stain, this is where the gram stain belongs. So you are using two stains. Some of the bacteria will take up one stain and other will take up another stain, so you can differentiate the bacteria. So there are in differential stains there are three other types. First one is gram stain, second one is acid fast stain, and then third one is Albert stain. In gram stain, the bacteria can be differentiated into gram positive and gram negative, whereas in case of acid fast, it can be differentiated into acid fast and non-acid fast group. In Albert stain, it will help you to differentiate between the bacteria who has metachromatic granules from the bacteria who does not have it. Okay. So this is a simple introduction to our staining techniques. Now we are moving on to our gram staining. So the first thing you have to do is you have to make a slide where your smear is there. You have to heat fix it, okay, because this is for bacteria. You're using heat fixation, so you are going to heat fix it. Now look at this picture. I have given there a white bacteria, okay. White bacteria is to show you that it has not taken up any color. Then it is put in a violet color solution, okay. So violet color solution will make you remember that the dye we are using here can be crystal violet, gentian violet or methyl violet. So what you have to do is that you have to keep it for 1 minute and then you are going to wash it off. So after this procedure, what happens, each and every bacteria will take up the color, okay. I have shown purple color inside their cytoplasm. So each and every one is taking up purple color, or sorry, violet color. Now what we are going to is, I am going to add a mordant. I am going to add something that will amplify the violet color. So what is the mordant here? The mordant here I am using here is the grams iodine. I'm going to use grams iodine. I'm going to wait for 1 minute. I'm washing it off. So what happens here is that the diiodine complex, there is a formation of diiodine complex, and this complex will become a little bit more big, okay. So see, to show you that iodine complex is bigger, I have shown a white bacteria with big granules of purple in violet or purple in color, okay. Now I'm going to remove the color, so I'm going to use decolorizer. So you can use acetone, you can use ethyl alcohol, or you can use acetone alcohol. Based on what you are using, your time also changes. So if you are using acetone, you have to keep it for 1 to 2 seconds. If you are using ethyl alcohol, 20 to 30 seconds. If you are using acetone alcohol, it's 10 seconds only, okay. So based on what you're using, your time will change. So after that, you have to wash off the decolorizer. You will get 2 types of bacteria here, okay. The gram negative bacteria will lose all the primary stain. The primary stain, what was the primary stain? Our violet color, right? Gentian violet, crystal violet or methyl violet. The gram negative bacteria will easily lose it, okay. But when you come to the gram positive bacteria, she is still having the granule, the diiodine complex in her. So she'll still be looking like a purple lady, okay. Now after that, you're going to use the counter stain. You're going to give another stain, okay, counter stain. So what you have to remember here that one group of bacteria lost the color, okay. So one is already having a color with them. So the one group which is already having color with them, they are not needing any more color, right? So who will take up the color? The one that is not having color right now. So the gram negative bacteria will take up the color of dilute carbonyl fusion and it will turn to pink or red in color, okay. So dilute carbonyl fusion, you have to keep it for 30 seconds. Is that okay? This is the simplest way to study the gram technique. So I'm going to just summarize it for you. So the primary stain, you use a crystal violet, 1 minute, then you will rinse it off. Then you're going to use a mordant to make a big diiodine complex. Grams iodine is the mordant that we use here. It is for 1 minute. Then you will rinse it off. Then you will use decolorizer, acetone for 2 to 3 seconds. Depends on what you are using, your time will change, right? Ethyl alcohol you have to use for 20 to 30 seconds. Rinse off immediately. You use a counter stain. So when you are using counter stain, gram negative bacteria will get the pink or red in color. Okay, now when you are asked in your viva, which is the crucial step? So the crucial step for grams procedure is decolorization. So decolorization is the most crucial step for gram staining. So if you're using decolorizer for more time, even the gram positive bacteria will lose color. So this is known as over decolorization. Or if you are using it for less time, gram negative bacteria will also do not lose the color. Okay, so it is under decolorization. So if you are asked in your viva, which is the most crucial step of the procedure, you have to say that it is the decolorization. So that's okay. Now we are moving on to the principle. So there are two principles that we commonly say for gram staining. One is that gram positive bacteria have more acidic cytoplasm. Okay, they are having more acidic cytoplasm, so they will take up the basic dye. Now iodine will act as a mordant and it will produce a bigger diiodine complex. But the most accepted theory here is the cell wall theory, where it is said that gram positive cell have thick peptidoglycan layer. So the peptidoglycan layer is not stained, but it acts as a barrier. Okay, now after the taking up of crystal violet, this will act as a barrier and will not allow the dye to move out. Also after using the alcohol, it will also shrink the pores. Again the dye cannot move out of the bacteria, and hence the stain remains in the bacteria itself. Whereas when it comes to the gram negative bacteria, there is a thin peptidoglycan layer, so easily the diiodine complex can pass through them, and also they are having a lipopolysaccharide layer which will get destroyed by the decolorizer, and it will produce large pores that will allow the diiodine complex to escape from the cytoplasm. So this is the very simple method to study the grams methods. Just remember the flow chart I have shown here, and just remember that the color of the solutions and the color that the bacteria takes up, you can easily remember how, and you can easily reproduce this in your exam. So if you liked my video, please do like, share and subscribe.